Hello, everybody, and welcome back to another ridiculously exciting episode of Puzzle Notes. This is an optional assignment, which means you do not have to do it. You do not have to answer the questions. If you do not do it and don't answer the questions, and I see a 0%, your score on this assignment will be your test score. If you do it, it's basically a free 100%. All you gotta do is take some notes and makes the questions pretty easy to answer. So you can take your pretty much free 100% on this assignment, or you can roll the dice and get your test score. Now some of you guys get 100% on basically all your tests, so you can go that way, or you can go the more for sure route. Anyways, selective breeding. I asked a couple students about this, and they were like, "What's selective breeding?" Well, that's what we did in class. That was what our critical reading article was about. And that's what our, try to make perfect puppies to catch birds activity was about. That is controlled breeding, where you're crossing two organisms in order to try and get certain traits in the offspring. With the dogs, we were trying to get perfect traits, like we wanted them to be fast, we didn't want them to be vicious. We didn't want them to be too strong. We wanted them to have good endurance, all that stuff. But in real life, you breed cows. You don't breed cows to make the least amount of milk. You try to get cows to make the maximum amount of milk. You don't breed chickens so they can lay one egg a year. No, you want them to lay as many eggs as possible, etc. So that's what selective breeding is. Selecting two organisms to breed in order to get offspring with desired traits. All right, so since this is quote unquote a review, since we should kind of know this stuff already, I'm going to go kind of quick. Remember, there are two types of selective breeding. The one type is called hybridization, or it's also called outbreeding. That is when you're breeding two individuals that are really, really different from each other. They're not similar at all. And you're hoping to have offspring that have the best traits from both parents. So that's kind of like what hold on. All right, I am back. So with outbreeding, you're trying to get kind of greedy. You're trying to cross two things in order to get the good from both parents. So kind of like a ridiculous example of this would be, like, say you grow tomatoes and you cross a tomato plant with, say, a cactus. And you want the offspring to not only make tomatoes, but you also want them to be like a cactus and not require much water. So that would be an extreme example of hybridization or outbreeding. Now remember, the advantages is what's most important. The advantages of outbreeding. If you're lucky and it works out right, you can get desirable qualities from the two parents. You can put them both together and you make we were trying to make the super dog. Another advantage. These offspring are usually healthier and larger. So, for the most part, if you spend like $2,000 on some purebred dog, they're not usually very healthy. They live, 7 or 8 years and then they die. But if you go to the pound and adopt a mutt, that's a hybrid, those things usually last 15, 16 years and they live for a long time. Another one is, with hybridization, since you're crossing two things that are very, very different, you're increasing genetic diversity, which in the wild is important, when we get to evolution, because with increased genetic diversity, you have a better chance for a population to survive and adapt over time if there's more diversity in the population. And now the disadvantages of hybridization or outbreeding. It is way more difficult to do. In my previous ridiculous example of trying to cross a tomato plant with a cactus, first of all, it probably won't work. Second of all, if it did work, your tomatoes might have spikes or thorns in it, which wouldn't be good. So it's really difficult to get, if you cross a tomato plant and a cactus, to get a really good tomatoes that don't require much water. You're probably going to get something you don't want. And in crossing members of two different species, almost always the offspring are going to be sterile. So if you ever wanted like a pet mule, well, you don't breed two mules, to get a baby mule. To get a mule, you got to breed a female horse with a male donkey. And that is how you get a mule. But two mules together, they cannot mate, they cannot have offspring. I know some of you guys saw the liger. That was a female tiger which was accidentally mated by a male lion. And I'm not 100% sure. I think the ligers may actually be fertile. They may actually be able to have baby ligers. But on the most part, when you cross two different species, they are sterile and cannot have babies of their own. Okay, now the other type of selective breeding, which is not outbreeding, but inbreeding. That's when you're crossing two similar organisms with very similar genotypes, which means they have very similar genes. These organisms are often related. So we're not talking about people here. We're talking about farm animals, plants, things like that. So the advantage of this is, first of all, it's much easier to do. It's much easier to get the desired traits you want when you're crossing two organisms that are very similar. An example of this is if you cross two purebred bulldogs, all the little puppies, they're all going to look like bulldogs. I mean, there may be tiny little variation. Some might be a little bigger, some might be a little smaller, but their colors are all going to be pretty much the same, and it's going to be kind of hard to tell one from the other. On the other hand, if you cross like two alley cats, like two stray cats, and they have 10 kittens, some might be orange, some might be black, some might be gray and white, who knows? You get a bunch of different things, but that would be outbreeding. But with inbreeding, you pretty much know what you're going to get. Another advantage is that the traits can actually be accentuated or enhanced over time. For example, like for dogs, poodles are a pretty big dog. Let's pretend like that. I don't want to draw a dog. Let's pretend like that circle's a dog, a poodle. Well, if you keep breeding the smallest puppies you got, they can keep getting smaller and smaller. Next thing you know, you have what's called a toy poodle, which is a pretty small poodle. But they kept even one of them smaller. They kept breeding smaller and smaller, and then you end up with what's called a teacup poodle. So with inbreeding, you can accentuate or enhance the trait you're looking for to get smaller and smaller and smaller, or bigger and bigger and bigger, or faster and faster and faster, whatever you're looking for. If you're looking for one trait, it's easier to enhance that trait. Disadvantages: harmful traits from the recessive alleles. Frequent inbreeding makes the offspring less healthy. They're more likely to have disease. And the opposite of outbreeding, genetic diversity or variability is reduced. So in wild populations, they have a decreased ability to adapt over time if there's some sort of environmental change. And that'll be in evolution. And just a picture that shows you how all the dog breeds have come about over, thousands of years. All dogs on Earth started as that, a gray wolf. And through selective breeding, they have turned into all these different breeds of dogs that do not even really resemble. I mean, this little guy came from that vicious wolf. I mean, that's crazy. But through selective breeding, the wolf turned into that. The little dachshund with his tiny little short legs and this fancy poodle. So selective breeding turned the wolf into all the breeds of dogs. And it doesn't really take too long. In 1790, which was just after the American Revolution, that is what the English bulldog looked like. And today it looks like that. So it looked pretty athletic back there in the late 1700s. And today, this very cute dog, but not very athletic. He's got the little short legs. He's very wrinkly. He gets tired if he runs like 10 feet. And these guys are not healthy at all due to inbreeding over several generations. And now on to real biotechnology. For thousands of years, humans have been trying to manipulate the DNA of plants and animals through selective breeding. and they still do it today because selective breeding is very cheap, but we do have the ability to now use genetic engineering. So we can actually skip the selective breeding part and go in to the organism's DNA and tinker with it. You can identify, change, remove, or add genes at the molecular level. So you can skip the selective breeding part. You can just go right into their DNA and get exactly what you want. So one thing you got to know, restriction enzymes. Remember, those are like the little scissors. They're made out of proteins, they're enzymes, and they cut DNA at precise locations. There are over 600 different types of restriction enzymes. They're naturally found in bacteria. You won't need to know that. But just know the restriction enzymes cut DNA at precise locations. And hopefully you kind of remember or have heard of this, gel electrophoresis. That is a technique that is used to separate the fragments of DNA once they have been cut by restriction enzymes. A little more on restriction enzymes. This is the one called EcoR1, which is like one of the most famous ones. And EcoR1 cuts DNA at an exact spot. It's looking for this specific sequence of GAATTC. Every time, and this little guy right here, that's the restriction enzyme. Every time it sees that exact sequence of bases in DNA, it cuts between the G and the A, and then cuts again between the G and an A. Now remember, whenever it leaves this little single strand at the end, that's called a sticky end. Sticky end, the TTA is going to go perfectly with the AAT. So those things can go right together, and then you have, once they come together, this foreign DNA, which has been spliced on to the natural DNA, you have what's called recombinant DNA. So recombinant DNA is DNA that has had genes inserted into it artificially in the lab. Okay, quick slide on gel electrophoresis. We did a computer simulation on this. The analysis part is also called RFLP, which stands for Restriction Fragment Length Polymorphisms. You won't need to know that. I'm not going to go over all that, but read that, and we'll see a picture on the next slide. And now a little picture of a gel electrophoresis chamber. Now remember, the first thing you got to do in gel electrophoresis is you cut the DNA using restriction enzymes. So let's just say, for example, we're going to use the restriction enzyme EcoR1. It's going to cut every time it sees GAATTC. Every time it sees that, it's going to cut and then cut like that. It's going to cut the DNA in half. But since everybody's DNA is unique, unless you're identical twins, then you have the same DNA. But other than that, everybody's DNA is different. So you're going to have the GAATTCs at different spots in your chromosomes, in your DNA. So let's say these are two different DNA samples from two different people. The restriction enzyme, EcoR1, is going to cut it at different spots. So let's say it cuts it there, and then way down there. And for this guy, we'll say it cuts it just there and there. So you're going to have fragments. You're going to have a little tiny little fragment up here. You're going to have a longer one, then a tiny one there for this person. This one you're going to have an average one, kind of long one, another average one. Then you're going to take that DNA and you're going to put it, now that it's been chopped up, you're going to put it into these wells. Now remember, DNA has a negative charge. That's because of the phosphate and the phosphate sugar backbone. Phosphate has a negative charge, therefore DNA has a negative charge. So now that we chopped up the DNA and put it into these wells, once we turn on the power source, the negatively charged DNA is going to be repelled by the negative end, and it's going to be attracted to the positive end. So very slowly, the DNA is going to start migrating from the negative side to the positive side. Well, as you can see here, going through this thick gel, The larger fragments of DNA like this, they have trouble traveling through the gel. They don't go very far. But the little tiny fragments like this guy right here, they fly through the gel. So they end up going woo, and they end up going way, way farther. So once you turn off the power source and you put it under UV light, you see these bands. And that is how you can compare two people's DNA, by how far the DNA travels. The farther they travel, the shorter the molecule. And the opposite of that, the shorter distance they travel, the longer the fragment of DNA is. So in a nutshell, gel electrophoresis is just a technique to separate fragments of DNA according to their length. All right, we already talked about this, but let's talk about it one more time. So what is recombinant DNA? Recombinant DNA is artificial DNA that is created by combining DNA sequences that would not normally occur together by splicing genes together in the lab. So when we did that little with the scissors and the tape where we cut out the human gene for making human growth hormone, and then we inserted it into the bacterial plasmid DNA, that was an example of how to make recombinant DNA, because the bacterial DNA now contained the human gene for making human growth hormone. Therefore, it is now called recombinant DNA. This all goes super fast because we did a whole project on it. Transgenic organisms. Hopefully you've at least heard of that after our project. This is where an organism contains DNA from another species which has been tinkered with through genetic engineering. Now, we really didn't talk about it, but hopefully you saw that to do this, these genes must be inserted when it is just a fertilized egg to actually change the DNA for the organism. The reason for that is, if you're a single egg, once you divide, that egg divides into, 2 cells, and then to 4 cells, and then into trillions of cells. All of those cells, if you put the DNA you want in that original cell, all of them, all the new cells will have that gene that you inserted into the original cell. You can't do it once you're an adult. So I know you have dreams, to have jellyfish DNA inserted into your cells so you can glow in the dark like a jellyfish can. Well, you can't do it because you have trillions of cells now. So you'd have to go in and insert that jellyfish DNA into all, 5 trillion or whatever cells you got. Well, you can't do that. You got to start when you're a single cell. So then when you grow up into an adult, all the trillions of cells will have that jellyfish gene to make you glow in the dark. Now, of course, they'll never do that with people, but they have done it with plants. And you can actually, I don't know why they don't sell these things, because I would definitely buy one. I would love to have a plant that glows in the dark, and maybe one day they'll have that for you to buy. All right, this slide basically tells you exactly what we did in that paper lab where we used tape and scissors. The bacterial transformation. Bacterial transformation. Basically, you have 2 first steps. First, you got to isolate the gene in human DNA for growth hormone. Then, right here, they're using the restriction enzyme EcoR1. I think in our lab it was HIN3, but it's EcoR1 here. I don't know what it is in real life. But they use EcoR1 to cut that gene, just the gene, because they don't want the whole chromosome. They just want that gene for human growth hormone. They cut it out using EcoR1. You can see here, it's got the little single strands on both sides. Those are the sticky ends. Also, at the same time, another step, you got to remove that plasmid, the circular DNA, from the bacterial cell. Now, that process is called curing. Well, you need to know that? No, you won't. But if you expose this bacteria to UV light, it'll actually eject its plasmid. In that process called curing, they're going to use the same restriction enzyme EcoR1 to cut open so they opened up that circular plasmid. It now has that nice space right there where we can insert the human growth hormone gene. With the right enzymes present, that gene will come right in, and the sticky ends will coincide. They'll have the same bases, so that gene will come right in and stick, just like here. So the gene for human growth hormone has been incorporated into the bacterial DNA, the plasmid. So remember, so now this would be recombinant DNA, since it has the gene for human DNA, human growth hormone, incorporated into the bacterial DNA that normally would not happen together. Now the only last step is to get that plasmid and put it back into the bacterial cell. Well, first, the bacterial cell needs to enter a stage called competence. Competence. And you do that by shocking it. Basically, you subject it to chemicals, which is calcium chloride. You will not need to know that. And then you heat shock it. When it does that, bacterial cells enter a stage. They become what's called competent. And when they do that, they will readily take back in exogenous or foreign DNA from the environment and include it inside of themselves. So now that bacterial cell, since it has the human DNA inserted into it, will now produce HGH, or human growth hormone. And the last thing we're going to go over is PCR, which stands for polymerase chain reaction. Hopefully you remember that the reason it's got the name polymerase is because of the enzyme DNA polymerase. Hopefully you remember from DNA replication and nucleic acids, there were basically two enzymes you had to know. You had to know helicase. That was the one that came through and unzipped. So it unzipped the DNA molecule. And then remember, it was DNA polymerase 3 that came along and attached the free-floating nucleotides to the exposed bases. Well, this is kind of like the same thing. Polymerase chain reaction, in a nutshell, is basically just a copy machine. So whenever I give you guys worksheets and I hand them all out to you, in case you didn't know, I don't type all of those up. I type up one, I print one, and then I go to a copy machine to make a bunch of copies. Well, that's what PCR is. But it's not making worksheets for you guys. It's making millions and millions of copies of DNA. Now, it does not use the enzyme helicase to unzip it, but it does use the enzyme polymerase to add the other side of the DNA. So this side hasn't been added yet. So this side's, still exposed, but they're going to add bases here, and we're adding bases here. So for PCR to work, first we need some DNA. We need the enzyme polymerase, and obviously if we're going to make a bunch of copies, we need the parts that make DNA. And those are, of course, the nucleotides. And a simple little diagram. And remember, you guys don't need to know exactly how this works, just the basics. I mean, obviously, the more you know, the more points I'm going to give you. But you don't need to become like a Harvard professor about PCR. But anyways, you start with DNA. Use what's called a thermocycler. That's basically like a thing that heats up and cools down. Heats up and cools down. Well, we don't use the enzyme helicase to unzip it. What we do is, what they do, I don't do it. They heat it up to almost boiling, 95 degrees Celsius. And when it does that, the DNA molecule unzips. So now these bases are exposed. It is ready to be replicated. These little red things, those are the primers. There's a lot of primers that are in there too. They rush in because once it cools down, if it wasn't for these primers that stick onto the DNA, that DNA molecule would simply close back up and it wouldn't replicate. But those primers rush in and stop it from just basically closing back up and being the same molecule again. Well, then DNA polymerase will then come and add the other side of this DNA, and it'll also come and add the other side of this. So we started with one, and after one cycle, we will have two, like that. And then you do it again. You will then heat it back up to 95 degrees Celsius. These two guys right here will open up. The primers will once again rush back in. DNA polymerase will come in and add it again. So as you keep going again and again and again, you just keep making more and more and more copies of the original DNA. And after 35 cycles, you can see you'll have 68 billion copies of that original strand of DNA. Alrighty, I had no clue this slide was on here. It's bacterial transformation again. Well, I guess if you didn't get it the first time, here we go one more time. And the first step is you got to isolate the gene for human growth hormone. Here they're using the restriction enzyme EcoR1 to cut it from the rest of the chromosome. At the same time, through the curing process, you're going to remove the plasmid from the bacterial cell. You're going to cut that plasmid again using the same restriction enzyme EcoR1, which leaves the little sticky ends. And you got this big space here that's now ready to have this human DNA inserted into it with the right enzymes present, especially DNA ligase. That DNA, that gene will be inserted right there. It'll be spliced in to the bacterial plasmid. And then remember, you got to make the bacterial cell competent. That's just a state. It's basically a state where they're ready to accept DNA from the environment, from the external environment, and incorporate it inside of its cell. If you get the bacterial cell to become competent, it will take in that plasmid, and then once again it will produce HGH, which can be used to treat people that need HGH. Thank you very much for listening. I hope you enjoyed it, and I'll talk to you next time.